Hello and welcome everyone. So as promised, today we'll be discussing about DNA methylation. Okay, so before learning about DNA methylation, let's do a quick recap of what we have learned till now. So first we learned about epigenetics, how this new field emerged and how the case studies like Dutch hunger winter and studying schizophrenic people and studying the identical twins inspired the researchers and they got an idea that there is something going on beyond the DNA sequence which is affecting the gene function. Okay, so I hope you all have learned about the theory of Lamarck that in that theory the Lamarck, when he studied the giraffes, and he said that the acquired traits which are means the traits which are acquired by an individuals, they are passed down from generation to generation. So at that point of time this theory was rejected, right? But epigenetics actually supports this theory. So what do we study in epigenetics? In the epigenetics we study about the changes in the gene functions which are inheritable, okay, which can be passed down from generation to generation mitotically or meiotically. But those changes in gene functions, they are not derived by the changes in the DNA sequences. Okay, so till high school level you have learned that changes in DNA means DNA mutation affects the gene functions. Okay, means if A gets converted to T or G gets converted to T, whatever DNA mutation is there will lead to different protein synthesis, and that will change overall the gene function. Remember sickle cell anemia? So this those diseases, those changes, those disorders are caused due to the changes in the DNA sequence. But due to those studies, due to Dutch hunger winter and all, scientists got to know that environment and your choices, like your lifestyle, your choices, whatever you are doing today, that can affect the gene function. Okay, so that's how a field of epigenetics emerged. And after that we learned about the chromatin architecture. Learning about chromatin is very important because understanding chromatin architectures helps us to decipher the basic molecular mechanisms in epigenetics. Okay, this like epigenetics, in the epigenetics we study the changes in gene functions, right? So how one cell or two different cells which have same DNA sequence can give two different outputs because they are reading the DNA differently. And why are they reading the DNA differently? Because of the difference in their epigenetic marks. And for learning about those epigenetics mark, how those modifications are done on chromatin, we'll first at least need to understand about the chromatin architecture, right? So we learned about the DNA packaging, how the long 6-meter long DNA is wrapped up around the histones and it is packaged inside the cell nuclei. Okay, so while learning chromatin architecture, we also learned that this chromatin architecture can be changed. It can be changed non-covalently or it can be changed covalently. So the non-covalent modifications in the chromatin, they are brought up by the chromatin remodelers. Okay, so how does that non-covalent modification is done? Because DNA histone interaction is changed. Okay, so the alteration of DNA histone interaction is done by complexes like SVSNF complexes. So this is the most famous example of chromatin remodeling complex. And then we learned about the mechanism. How does it bring upon the change in chromatin structure using hydrolyzing ATP? So we learned about all those. And then other than non-covalent modifications, means other than altering DNA histone interaction non-covalently, there is the other type of modification which is covalent. So those covalent modifications are done on the histone tails or histone cores, and they are done by the histone modifiers. Okay, so we learned about the highly conserved domain of which is known as histone fold domain in the histones. While remember, while learning about nucleosomes, while learning about octamer, we learned that histones have highly conserved domain which is known as histone fold domain, and alongside this domain they have C-terminal extension and as well as N-terminal tails. So those N-terminal tails, this polypeptide stretch at the N terminal of the histone is very important because at this on those amino acids, all of the modifications are brought up which are going to interrupt the chromatin architecture. Okay, so those modifications are done by the histone modifiers. So those modifications are methylation, acetylation, means addition of any chemical group by certain enzyme. Okay, so those modifications are covalent modifications, but those all of those are post translational modifications. Post translational means after those modifications, they are which are done after the translation, means those all of those modifications are done on the proteins. Those are the changes that are brought up in the proteins after they are synthesized. So means histone tail modification or histone core modification, all of those comes under the histone all of the comes under the post translational modification. But the DNA can also be directly modified. Okay, so DNA can be directly modified by addition of certain groups, certain chemical groups, and mostly 80% of the times it is the DNA methylation. Okay, so DNA methylation can be a very important modification. So in the epigenetics, whenever we learn about the modifications, those are post translational modifications, but those post translational modifications are done on the histones, on the proteins. So that's why we call them post translational, means after the translation. But the modifications can be done on the DNA too. And the most frequent, the most common modification is DNA methylation. So as the methylation term suggests, that it is the addition of CH3 group on the DNA. And the take home message for you all is that DNA methylation always lead to the silencing of gene. So whenever there is DNA methylation, there will be repression of the gene. Okay, so gene will be silenced, and it will means it just switch off the gene. Okay, DNA methylation switches off the gene. So let's learn about the pattern of the DNA methylation. So on the DNA, methylation is not done on anywhere. Okay, so it is always done on cytosine. But for cytosine to be methylated, there is a prerequisite. So what is that prerequisite? Let's assume that this is the DNA sequence. Okay, so this is some DNA sequence. So I told you that cytosines on the DNA are methylated, means CH3 group is added on the cytosine. So there are two cytosines present, this and this. But for this cytosine to get methylated, there is a prerequisite that this cytosine should be preceded by a guanine. Means if there is cytosine and it is followed by guanine, only this cytosine will be methylated. No other cytosine than that will be methylated. So those dinucleotide C and G dinucleotides are often written like this: CPG, means C, phosphodiester bond, and G. So this is the conventional way of writing CG dinucleotide. So we call it CPG. So whenever there is CPG, that C will be methylated. But whenever the C is not preceded by, whenever C is not preceded by G, then that cytosine will not be methylated. Okay. So CPG islands, CPG dinucleotides are methylated. So this is the pattern of the methylation. I hope that's clear to you all. So here you can see this is the cytosine molecule, and on the cytosine molecule at the C5, at the fifth carbon, the addition CH3 group is added, and cytosine is converted to 5-methyl cytosine. Cytosine C is converted to 5-methyl cytosine. Okay, so this conversion is done by using the enzyme known as DNA methyl transferase. So, as the name suggests, methyl transferase, the enzymes which ASE always denotes enzyme. So the enzyme which is transferring methyl group on DNA. So whenever there is something which is used as prefix, it will be always the name of the substrate. Means this substrate means the substrate on which the enzyme is going to act. So this enzyme is going to act on DNA. That's why we call it DNA methyl transferases. So it is a transferase, means it is transferring methyl on DNA. So DNA methyl transferases transfer methyl on the cytosine and convert it from the cytosine to 5-methyl cytosine. So from where do those DNA methyl transferases get CH3? So donor of the CH3 group, donor of CH3 group is S-adenosyl methionine, okay, which is also known as SAM, S-adenosyl methionine, and it is also known as adomate. Okay, it is also called as adomate. And after donation of a methyl group, S-adenosyl methionine gets converted into S-adenosyl homocysteine. Okay, SAH, S-adenosyl homocysteine, and it is also written as ADO-HCY, means adenosyl homocysteine. Okay, so SAM gets converted to SAH. So SAM donates the CH3, and that CH3 group is transferred on cytosine by using DNA methyl transferase, and that is converted into 5-methyl cytosine. Okay, so you already know that cytosine can undergo deamination and get converted to uracil. Okay, so this is the cytosines and uracils. Those are present in RNA, so that it can differentiate between the cytosines. Okay, so the deamination of cytosine converts the cytosine to uracil. Well, if the deamination, so if the 5-methyl cytosine undergoes deamination, it gets converted to thymine. Okay, and that is why the methylated DNA is highly vulnerable for mutation, because the 5-methyl cytosine can be easily deaminated and converted to thymine. Okay, so we learned that the DNA methylation is always done on the CpG dinucleotides. Okay, so 80% of the methylation of the DNA methylation, it is done on CpG dinucleotides. Okay, so those CpG dinucleotides, they are not present equidistantly. Okay, the distribution is not uniform. So what happens that CpG dinucleotides are present in clusters. Okay, so let's say here there are 50 to 60 CpG dinucleotides. Okay, and let's say here there are 600 to 800 CpG dinucleotides. Okay, so this is how the CpG dinucleotides are present in clusters, and those clusters of CpG are known as CpG islands. Okay, those are known as CpG islands. So there are two types of islands. First, which are CpG poor islands, which has CpG frequency, but it is comparatively low, while other are CpG rich islands. Okay, so there are two types of islands: CpG poor islands and CpG rich islands. Here, CpG rich islands and CpG poor islands. So those islands vary in terms, they vary in terms of a lot of different aspects. So let's have a look at the differences between CpG rich island and CpG poor islands. So let's compare both of them. So first, understand how methylation affects those ions. So we learned that methylation can be done on CpG dinucleotides. So no matter whether they are rich in CpG or they are poor in CpG, those will be methylated. Okay, but the methylation frequency, okay, those that will be infrequent on the CpG poor islands, CpG rich islands, methylation will be infrequent, while in the CpG poor islands, methylation will be rich, or say more frequent. So you could say that CpG rich islands are poorly methylated, while CpG poor islands are heavily methylated or highly methylated. Okay, so you can say that this is vice versa. Frequency of the methylation is vice versa. If CpG frequency is less, then the methylation will be higher, and if the CpG frequency is more, then methylation will be poor. Okay, so this is about CpG frequency. Okay, let's learn about the just let me erase this. So this is about the frequency of methylation. Okay, this is about frequency of methylation. Let's learn about the effect of methylation. What is the effect of the methylation on both of those? So when even though the CpG rich islands have infrequent or poor methylation, but whenever there is methylation, there will be immediate silencing of gene. Okay, but on the CpG poor islands, which are heavily methylated, the methylation kind of has indefinite effect. Okay, what does this mean? That it won't immediately silence the gene. For silencing of the gene in such cases on the CpG poor islands, you need to look at the other factors too, like the enhancer regions, whether there is presence of enhancer, what is the availability of transcription factors, and again what is the status of histone modification. Okay, whether the histone modifications are activating modifications or repressing modifications, and how is the chromatin architecture, whether it is open or it is in closed configuration. So all those factors plus methylation, DNA methylation decides whether the gene will be activated or repressed. Okay, so like DNA methylation is not the only factor which is going to control the expression of the gene on the CpG poor islands. But if there are the CpG rich islands and they are methylated, and for example, this is the gene. Okay, this is gene X, and the promoter of this gene, this gets methylated. Okay, this is how the CpG dinucleotide methylation is shown. If this is like this without shading, it is known as unmethylated CpG islands, and if it is shaded, this is methylated CpG islands. Okay, so if the, let's say this upstream of the gene X, or the promoter of the gene X, methylation is done, and this island, this CpG island is CpG rich island. Okay, and if this CpG rich island is methylated, the gene X will be immediately silenced, and this will be 100% for sure, it will be silenced. But if those are CpG poor island, instead of CpG rich island, we have CpG poor island, and it is methylated. Now, this is not the only deciding factor. You will have to look at the if there is a presence of enhancer region, what is the availability of transcription factor, what is the status of histone modification. You will have to look at all those different factors alongside the DNA methylation, and now you will get the status of the gene, whether gene is in the on state or gene is in the off state. Okay, so I hope this is clear. So what happens is rich islands, in the CpG rich islands, if they are methylated, the gene will be silent. So, but in the CpG poor islands, if the gene if those CpG dinucleotides are methylated, it can be silent or it cannot be. Okay, it depends on the other factors as well. This is the effect. Now let me ask you a question, that there are two types of genes: housekeeping genes and genes and tissue specific genes. What are housekeeping genes? So housekeeping genes are the genes which are constitutively expressed, means these are the essential for the survival of cell. So no matter what the cell type is, no matter at what stage the cell is, means whether it is differentiated or whether it is not differentiated. So all those cell stages as well as all the cell types need the expression of those genes. So such genes are known as housekeeping genes. For example, ribosomal genes. Okay, so ribosomes are protein synthesizing machinery, and they are very essential for protein synthesis. So tell me single cell which doesn't require protein synthesis, right? Every cell type needs protein synthesis, as well as whether it is embryonic cell, whether it is pluripotent cell, or whether it is differentiated cell. No matter what the cell stage is, all of the cells, all of the cell types needs those genes. So such genes are known as housekeeping genes. Well, some genes are required in certain tissues, while they are not required in other tissues. Such genes are known as tissue specific genes. So my question is that which kind of islands will be found at the promoters of those genes? Means at the promoter of housekeeping genes, whether we'll find the CPG rich island or CPG poor island, and at the promoter of tissue specific gene, whether we will find CPG poor islands or CPG rich island. Okay, this is the question. So pause the video and think about it. Okay, because I know I guess explaining the effect and methylation frequency, this should be clear to you all, but still think about it. Okay, so so housekeeping genes are essential, so they are required in all of the cell types. Well, tissue specific genes, they are required in certain tissues, right? Okay, let me explain you. Housekeeping genes, the promoters of housekeeping genes has CPG rich islands, while the promoters of tissue specific genes, those have CPG poor islands. Okay, why is this case? Because at all those housekeeping genes, they are constitutively expressed, so you don't really need an epigenetic switch which will be switching on or off the gene, and that is why the those genes have CPG rich island. Means there is only one deciding factor whether the gene is switched off or switched on. But generally, those are constitutively expressed, so they will be showing high expression at any cell stage and in the all cell types, and that is why the methylation at methylation frequency is infrequent. The CPG rich islands are poorly frequented. Why? Because those islands are always found at the promoters of housekeeping genes. So now tell me, if the housekeeping gene promoter gets methylated, and it will lead to, what will it lead to? It will lead to the gene repression. So if you switch off the housekeeping gene, means you are switching off the gene which is very essential for you. How the cell will be functioning now, right? It is kind of cell suicide. Okay, you are telling the cell that don't produce something which is very important for your living. So that is why the methylation frequency at CPG rich island is very poor. Those are poorly methylated islands, and those, why? Because they are found at the housekeeping gene promoters. Well, on the other hand, tissue specific genes, here you need an epigenetic switch which can switch on and off the gene depending on the requirement. Okay, if the certain tissue requires that gene, you need to switch on the gene, and for that you will not just, not just methylation decides, but the other factors also decides whether the gene will be switched on and off. And that is why those not the frequency of the methylation is very high, because because there are a lot of genes which are present in the cell, but they are not required for that cell, but they are required in some other cell. For example, let's take the dopamine gene. Dopamine gene is required in neurons because it is a neurotransmitter, but it is not required in the muscle cells, right? What will muscle cell do with those neurotransmitters? So depending on the requirement, those can be switched on and off. So those tissue specific genes, they have the CPG poor islands, and that's why CPG poor islands are frequently methylated. They are highly methylated because methylation do not directly lead to the gene silencing. Of course it does, but along with the DNA methylation, there are requirement of the other deciding factors which are going to decide whether the gene will be expressed or not. So I hope this is clear to you all. So let's now take a look at the examples. So genes like MAGE genes, MAGE genes. So these are found in both somatic cells as well as germ cells. Okay, but those are only required in germ cells. present, they are present in both of the cells, but only required in germ cells. So because they are required in germ cells, okay, now, so I forgot to tell the most important thing, that MAGE gene promoters have, they have CPG rich islands. Okay, because I've written it here, so it's by convention that those are they have CPG rich islands. Okay, so those genes, they are present in both somatic cell as well as germ cells, but in the germ cells they are required, means they should show the expression. Okay, so they should be in the on state, and that's why in the germ cells they are not methylated. The islands, the CPG rich islands, are not methylated because you don't want them to switch off, right? But in the somatic cell, they are the genes are present, okay, but you don't need the expression of those genes, and that is why the CPG rich islands at those genes are methylated. Okay, why? Because the DNA methylation on those CPG rich islands are going to lead to silencing of the gene. So this is how the methylation on the CPG rich islands is, it is altering the gene, it is altering the gene function, because at the developmental stages, somatic, at the start, somatic cells and germ cells, there is no differentiation. Like cell has not committed that, okay, I will be somatic cell or I will be the germ cell. So switching on and off of certain gene that decides, okay, this will be the cell fit. This cell is going to be the cell of epidermis. This cell is going to be the cell of mesoderm. That's all. That commitment is decided. And while this commitment, you need to switch on and off the genes, and that is why the somatic cells, MAGE genes, are methylated, because you do not require MAGE genes in the somatic cells. So what happens that in the when certain things go wrong, okay, for let's say that there is certain gene X, and it needs to be silenced in all of the cells, in all of the normal cells, okay, but due to certain condition it doesn't get silenced. Means, say this is the gene X, okay, and this is the promoter of gene X, and this promoter of gene X is methylated, okay, and that is why the gene X is silenced. Okay, this is why the gene X is silenced. And but due to certain conditions, due to the deficiency of vitamins, like folic acid and vitamin B12, vitamin B6, this gene doesn't get methylated. Okay, and due to hypomethylation of this gene, the gene X get activated, and this can lead to cancer. Okay, this is how the disorders are, this is how the individual suffers from different disorders. Okay, this is how methylation can affect the gene function, and it can lead to different disorders. So MAGE genes are silenced in somatic cells because of the methylation, because of the methylation of CPG rich islands. So there are certain DNA demethylating agents. Okay, let me write it in a different color. DNA demethylating agents means, as the name suggests, those agents are going to demethylate the DNA. Okay, so the example is 5-azacitidine. So those DNA demethylating agents, what do? They demethylate the DNA which was already methylated. So let me ask you, if you put this 5-azacitidine in both of the in the somatic cell and germ cell, what will happen? When you put 5-azacitidine on the somatic cell, it will demethylate the DNA. So this methylated DNA will be demethylated, and this, due to demethylation in the somatic cell, the expression of MAGE gene will be seen, right? It will start producing the proteins it is designated to produce. So but you don't need there is no requirement of the expression in somatic cell, and this leads to the cancer. Okay, so this is how methylation can have huge impact on your health. Okay, there is another example. There are the transcription factors like OCT4 and Nanog. Okay, those are the transcription factor, which are, these are the markers of the undifferentiated cell, means those are the transcription factor which helps in maintaining the pluripotency. So the all of the undifferentiated cells have those transcription factors. So what happens that during the development, during early development, the cells are in a pluripotent cell, right? Pluripotent state, and after that they need to be differentiated. So what happens that those genes have CPG poor islands. Okay, those have CPG poor islands, and those CPG poor islands, they show limited methylation till the epiblast stage of the early development. This is the epiblast is the stage of the early development, after which the pluripotency is gone, and actually cells start to march further to the lineage commitment. Means before the epiblast stage, means the at the early development, before the epiblast stage, there is limited methylation. Okay, so methylation at CPG poor island won't be leading to repression. Okay, I told you the other factors are also important for the deciding that whether this gene will be switched off or on, and that is why the limited methylation will be there. But after the epiblast stage, methylation starts to increase. Okay, so there you can see high methylation after the epiblast stage. After epiblast stage, why? Because after epiblast stage, cells are marching toward the lineage commitment, means like they are losing their pluripotency and they are deciding that they are going to be this or this cell type or that cell type in the individual. And this is what we call as lineage commitment, that they are committing to either a neuronal lineage or they are committing to either muscle lineage. This commitment, for this commitment, they need to lose their pluripotency, so they need to lose those transcription factor which are required for maintaining the pluripotency. And what do they do then? They just silence the gene which are going to produce those transcription factor, and for the silencing you need to increase the methylation. So before epiblast stage, you see the limited methylation, but as the individual develops further after the late developmental stages, the methylation of these OCT4 and Nanog genes is seen to be increased. So this is it for today's class. In the next class, we'll learn about the effect of DNA methylation on chromatin architecture. Like how, for now, we just learned about the functional role, right? That DNA methylation will be leading to repression of gene, but how it actually affects the chromatin architecture. We will learn about that in the next lecture. We'll also learn about the DNA methyl transferases, the types of DNA methyl transferases, and how do they function. So this is all for today. And in next class, let's meet with a very, very interesting topic, and that is DNA methyl transferase. Till then, see you. Bye bye.